science talk series here at the SeaWorld Research and Rescue Center. And I'm Dr. Elise Marquis, and I'm welcoming you to the center. As you know, most probably this initiative is carried out in partnership with the Environment Agency Abu Dhabi, Emirates Natural History Group Abu Dhabi, the Natural History Museum Abu Dhabi, and Nautica Environmental Associates. So I will thank you again for their presence and for their support. As some of you may know, plankton ecology is my research background. Actually, it all started during my master thesis. I had the opportunity to be part of a study on the planktonic food web of the Bay of Biscay on the west coast of France. And this was one of the best things that happened in my life, because this all came to me being here tonight, really. It did open an entire new world for me. I didn't care so much about plankton before that, but then I really discovered the wonder of the planktonic world. And with that, I had the chance to fulfill my dream of adventure on the ocean. The large extent of the ocean was just becoming my playground as I was studying and looking for those little guys here for many years in the Bay of Biscay. These are part of the plankton, microzooplankton. They are ciliates, unicellular organisms that are super important for the planktonic food web, for the carbon cycling, for the fisheries, and for the entire ecosystem. So I am super excited, obviously, about the topic of tonight, plankton, microbes of the sea. I had to fight with myself to not start the whole series with that topic. So I'm kind of glad that I could put it early enough in the year, in the new year. And I'm not gonna talk so much about it tonight, but I do hope that we will have more talk. If you enjoy the one tonight, we will be programming or scheduling more talk about plankton, I do hope. And I'm very glad to not be on stage tonight, but I will leave the stage to wonderful experts here with us, Dr. William Wakalevu, and Dr. Shadia Amin. They will be talking about their research and about their experience. Just remember to keep your question for the end. We will have the Q&A session at the end. So just listen and then ask all the questions you want at the end. All right. Dr. William Wakalevu. I just want to introduce him first. Willy was the first one to join the first researcher to join the research team. So we've been working together for 2 years now, and this is with a great pleasure that I'm calling him on stage. Willy, before coming here in Abu Dhabi, had many lives, starting from his island in the Pacific Ocean, going through Japan and ending up here in Abu Dhabi. We are very lucky to have him in the team. He's been working on many topics, mangroves included, and now he's leading the aquaculture research program for the center. But tonight he will be focusing on plankton. So here, to you, Willy. Thank you.
All right. Good evening, everybody. My name is Willy. I am from Fiji, very long way from here. Thank you very much for giving your time, precious time in the evening, to come and listen in on a talk about plankton. So, marine drifters and unsung heroes, the Rambos of the ocean. Usually I begin my talk by giving the definition of plankton. Before I do that, Who here has gone swimming in the ocean and has had a mouthful of seawater? Almost everybody. Well, guess what, folks? You may have swallowed some of them. They come in different sizes, they come in different shapes, they come in different colors. Be thankful that you swallowed the non-toxic ones. So I have here, which we will pass around, bottles of some plankton that you can have a look at that we grow in-house. These are zooplankton, which I will discuss a little later today. So as I go on in the progress through the presentation, please have a look at the plankton. You might have to hold them up to the light. I do not recommend drinking. So, definitions. Plankton. So in today's age of technology, we usually go for the shortcut. We usually Google what is plankton. So what's the usual first hit? Wikipedia. What does Wikipedia tell us? It says, small organisms suspended in the water, neither attached to the bottom or are able to swim effectively against a current. Small, suspended in the water column, not attached to the bottom, can swim against the current. The very next sources would be like encyclopedias, a little bit more credible. Now, they go on and say, marine and freshwater. So it's not only in marine ecosystem or marine environments, it's also in freshwater. Many do not know this. They're also in soil, and they're also in the air. So if someone sneezes on this side, please don't sneeze. It travels. It doesn't choose a nostril to go up. It just travels with the wind, going with the same definition. And Merriam-Webster says passively floating, but possibly weakly swimming. So there is some capability to swim. Victor Hansen, late 19th century oceanographer, very early stage oceanographer and microbiologist. He is the gentleman up there on the top left, or right, sorry. He mentions them as minute drifting animals. Now he's added on an additional, also plants. So they're not only small or attached to the bottom, swim effective against currents, they cannot do that. They're not marine freshwater, now we know soil and air. They not only passively float, chilling on the currents, but they also can swim. And they are also plants. And he mentions, imagine this is the late 1800s. They're the lifeblood of the sea. So, the term plankton comes from an ancient Greek word. I believe our second speaker, I had a peek at the slide and I saw him defining it as well. I'm going to get ahead of it. It comes from haliplankton. So haline, referring to the marine or the ocean. And plankton, plancel, Greek word meaning a drifter. So like an oceanic drifter, which fits the definitions that we have here. So, types of plankton. Now we know what they are. Let's have a look at the types of plankton very generally. There are two types, holoplankton and meroplankton. So holoplankton are organisms that spend their entire life. They may be a cyst, an egg, but when they become an adult, they still match or fit the definitions that we've shown before. These include diatoms, dinoflagellates, microalgae, some copepods, even they are gelatinous zooplankton. You'll be surprised later on about some of this gelatinous plankton. Now, meroplankton, how are they different from holoplankton? These are organisms that are only planktonic for a part of their life, not their entire life cycle. So they may be an egg or a larva only as plankton, and then they graduate to become part of the necton. So the difference between a plankton and the necton are able, or fishes that are able to swim against the current. So you have plankton or benthic existence, like crustaceans. Benthic meaning benthos or substrate, ocean bed, seabed, ocean floor. Who can tell me what this really looks like? A bit like an alien, doesn't it? This plankton is. Can anyone tell me? Please don't talk Rahim or Alex, my team, because you probably know what this is. or Mike Price. Or this one. See, look, this one's got a lot of spines. This is an adaptation. If you're small, you want to look bad because you want nothing big to eat you. So look at all those spines. You wouldn't want to eat something with spines like that. And look, this is called Paralichthys olivaceus. It's the olive flounder. It starts off normal, like any other fish. Eye on this side and eye on this side. After 16 to 20 days, it begins to go sideways. The eye migrates from one side to the other side. All the organs rearrange. That's pretty cool. And then you have sea urchins. Look at that stage of the sea urchin. Looks more like a NASA shuttle. And these are the adult forms. So you have here, this is the life cycle of a Scyllacerata, a very common crab, mud crab. You see them at Lulu. Delicious. You also have here the adult size of Paralichthys olivaceus, the olive flounder. They get up to 30 centimeters or much bigger. So different names. You have the flounder, halibut, flatfish. So, as I was mentioning, they start off with eyes on both sides, and then it migrates to the top of the head. And then, of course, a globular urchin. I better speed this along because I got 25 minutes. Two main categories of plankton, phytoplankton and zooplankton. There's, of course, multitudes of still unknown plankton. Bacteria, as our second speaker will allude to. So generally, I'll speak on phytoplankton and zooplankton. Photosynthetic unicellular protists and bacteria, they are the basis of the food chain. Everything eats up from them. They are microscopic, around femto to micro in size. And they are autotrophs. So they make their own food. Sunlight's all they need, a little bit of carbon dioxide, and they're good. Heterotrophic plankton, which is zooplankton, these are animals. They require to eat other things in order to gain their energy. And some of them are detrivorous. Things die in the ocean, where do they go? They sink, as marines know. Other planktons feed on this. So zooplankton include the meroplankton, where some parts of their life, larvae. So these include fish, crustacean, eggs, and larvae. How big are plankton? Now, you see the big red line at the bottom? It kind of throws off the definition, doesn't it? We were going minute, small. It goes above 20 centimeters. So you start with viruses, femto. The human eye, as you pass around that bottle, can see, the naked eye can see around 20 micron. Looks like a speck of dust. Anything below that, you need a microscope. So, as you go down this table, they get progressively larger. Now you have siphonophores, jellies, and some very interesting siphonophores. There we have a look. Bam. Those are also plankton. You see this? This guy has been found to be the longest living organisms on the planet, not the blue whale. It's a plankton. And I just got in my excitement, I pumped it ahead. So it can grow up to 45 meters, but they grow in the darker regions of the water. So you have sunlight, you have twilight, you have midnight, and abyssal. So around 1,000 meters depth. That's where this guy is found. And then you have this gentleman here. He's a jelly. True jellyfish. They can go about 30 centimeters to 5 meters. No, not 5 meters, 2 meters. So these guys, the definition is a little bit loose there on the size, but it meets the traditional definition of its drifting. Ecological importance. So we were just discussing, just before the talk, no plankton, no life. The importance of plankton. They're usually, that's why I put in the first slide, unsung hero. They're often underappreciated. So let's look at the marine food web. That's a very traditional food pyramid, beginning with the basis phytoplankton, the autotrophs, going on to zooplankton, small fish and big fish, and then, apex, right at the top. Then you also have the confusers, that you have such big bodies that are also eating plankton. things like as big as we have in our display, the manta ray. We also have whale sharks. These are all planktivorous, so huge, but they require such a large biomass of plankton to keep alive. As alluded to by Dr. Elise, they also play a very critical role in the carbon cycle. Right now, it's a very hot topic in the last decade or so, climate change, CO2 levels. Just to their sheer size, plankton are very effective, very efficient carbon sink. And I forgot to mention primary producers, which means they generate oxygen. They chew up that carbon dioxide with the sunlight and they produce oxygen. Studies have shown 50% of the oxygen that we're breathing right now is from phytoplankton. So every second breath, phytoplankton. There are also bioindicators. So they show seasonal cycles. These are pictures from NASA, not mine, I didn't go to space. Eutrophication. So if you're not familiar with eutrophication, if you, for example, use fertilizer to assist your crops up in the river systems, it rains, it drains the soil into the river, carries down to the sea, and fertilizer, they are plants. It's like chocolate for these guys. They love it, and they bloom. So the other downside is also they can be the toxic ones, variety which give food poisoning because they bioaccumulate. You have one little particle on a seagrass, it's being eaten by another small fish, a bigger fish, and then we get in there. Some examples are like ciguatera poisoning. I've had ciguatera poisoning. I don't recommend it. Biodiversity. They expand geographic distributions. So if you have fish that are spawning here and they drift with the current, they wouldn't be able to actively swim there. The currents take them there. So what would either be localized here is now elsewhere. Genetic diversity as well. Also, many don't link fisheries, economies, and livelihoods to plankton. We have these vertical migrations, which are critical also to understand fisheries. Because where plankton are, there's also fish. And where there's fish, there's fishermen. Here we have a picture, a graph showing at night, in the day, and at night. Plankton masses going down. It's called diel vertical migration. In the evening, in the daytime, they are down here. At night, they go up and feed on the surface waters. Obviously, ideally, you don't want to be a small guy up in the surface in the sun when there are bigger fish who can see you clearly from the bottom. So that's why they do these vertical migrations. Upwelling zones. Again, like the economies of Peru and Chile that are so reliant on heavy fisheries for anchovy and sardines. These are reliant on the upwelling. When the cold North Atlantic deep water comes through the ocean bed, hits the continent of South America, rises, nutrient-rich, fisheries bloom, economies thrive, money in the pocket, livelihoods improve. So you can see a chain of effects from the very small microscopic organism that is plankton to livelihoods. There's a sort of link. Studying plankton. Feel free to come and have a look after. Please don't break because it's pretty expensive and it's not mine. Companies have made business out of building equipment that are meant for sampling plankton, such as the Danish company KC Denmark. You have different types. Pumps, ones that run along the seabed and you just tow it. And the plankton collects up there in the collector box. You have vertical ones that go like this, that sample all the way up. And then you have the ones that lie horizontally, like these ones, that does the Newton or the stop surface water. And then you have traps that are suspended like this, or traps that are attached to the substrate. Right? So, and some are rigged on ships. All of that rigged on one ship. I've had the pleasure of working with some of the Japanese researchers studying the Kuroshio current, which comes in from Southeast China Sea through the Tokara Strait and up into the Northwest Pacific. This is what it looks like on a rosette, all set up. And here we are just trying before deployment. And it goes down all the way to around 250 to 500 meters, sampling randomly all the way down to understand the strata, right? So, I have 3 minutes, and I'm going to fly through it. There's the map, drifting with plankton. This is my life story. So just like plankton, I drifted almost to the far corners of the world, literally studying plankton as I went along. So there we are in the red circle, UAE. These are some places where I've worked. Some of you may have not heard of these places. So of course, Japan, where I worked with the Japanese government and the Taiwanese government, studying the Kuroshio current and the Kuroshio paradox. Solomon Islands, if you remember, it's just east of Papua New Guinea, northeast of Australia, working on nurseries, understanding biodiversity, on how these planktons use seagrass and mangroves as nursery habitats. Fiji, understanding delta areas and the richness, species richness and abundance in estuarine systems using light traps. And French Polynesia. Many people don't know much about French Polynesia, just Bora Bora. Understanding coral reef fish larvae behavior prior to the return to the reef. So I'll just fly through this. Yeah? Really? Guys, get ready. It's going to take an hour. Coral reef fish reproduction. To understand coral reef fish reproduction, I'm going to have to divert a little bit into explaining a little bit about the behavior. So 56% of coral reef fish, they broadcast spawn in the water column. So eggs and sperm in the water column, and then they fertilize externally. And this is usually done at the time of month, moon, tide, dependent. 6 to 8 weeks, these guys are out in the open ocean. It's pretty cool. If you remember from one of the very first science talks, Dr. Gerrit Naninga alluded to how these fish return. So I'm going to talk about the section that's in between. 6 to 8 weeks, some of them, without being told, return to the very reef they were spawned, to the very coral head. That's pretty cool. No one told them, oh, that's the one that your father was. That's pretty cool, though. So you have the different zones of the reef. 6 to 8 weeks as plankton, remember, meroplankton. Then they come and cross the reef, and they call it colonization and settlement before they grow into adulthood, and again the cycle begins. So the study was done in 2 very different localities in French Polynesia, a high volcanic island and an atoll. Who here has been on an atoll? All right, which atoll, if I may ask? Oh, in the Maldives, okay, other side. Okay, so Nango Nango Atoll, it's like a donut. It's a very top, if you imagine it's a very top of a mountain. The rest of the body is underwater. So in some areas, you can pick a rock, throw it, hits the water that side, you pick the rock, it hits the water this side. That's how thin the land is. And Moorea on Society Archipelago. Reference, okay. So these were the equipment used. Light traps, which were suspended outside the reef in the open ocean. Crest nets, which are fixed here, and a horn net, which is, when you have a donut of land, there are sections where continual replenishment of seawater comes in. And this net, its goal is to capture the larvae as they are returning to the reef. This is a video of the installation, very short. So you see that's the mouth, open oceans on the other side. While you watch the video, if you've this place is an abandoned pearl farm. There were only 3 of us and 2 crazy scientists. My supervisor put us in this in the middle of nowhere. If you don't fish, then it's petit pois for you. So I'm not much a big fan of petit pois. And if your boat malfunctioned, let's hope you hit Galapagos. All right, so all traps were set before sunset. At dawn, they're collected. They're all ID'd, counted, sorted, and kept in flow-through cages. So that if it's hard to identify, for example, lobster larvae, you can grow them up in the cage and see what they end up to be, so for correct ID. and sort of in this idea, also at different depths. So just the main part of the results, there is a lunar preference. If you look at this graph, this is new moon, that's a full moon. At new moon, there's a bulk of settlement. So it's common sense also. So if you're very small, you're clear, they still can't see you, but you don't want to be hanging around when there's a full beam of light on you. You want to hang out, chill a minute, until it's a little bit darker, then you can cross. If you imagine this as the reef, this is the seabed, steady climb, surf zone, and where the cupcakes were is where you need to be. I need to get to the cupcakes. But I have all this wall of eyes looking at me, waiting from the other side. These are adults, right? 90% of fish larvae die. They survive the open ocean 6 to 8 weeks, then they got to contend with all the wall of eyes looking. So, very small percentage make it through. So, around new moon nights. And it's also depth dependent. They don't want to be hanging on the surface until they're really close to the reef crest. So, I don't want to be hanging on the surface from here because all the big fish can see me. As soon as I get close to the cupcakes, pop over, use the waves, crest through. And it's the same pattern of colonization. So it's groups. So a dozen of villies will just, okay, guys, let's go. I don't know, I'm not ready. And then they cross. It's usually in patterns of species like this. You get your convict triggers, convict surgeons. They all hang around, and then they go together. Numbers counts. Not good for the individual. And environmental cues. So again, Garrett spoke about this, so I'm not going to go into detail about that. And some of this work was published in PLOS and Fish Biology. So here, this is my last slide, at least. You can breathe easy now. I can stop waffling. So what do we do? What are we planning to do also here? So these are some of the equipment that has been acquired by the center so that we can conduct some of these similar studies here in the Gulf. We've already got some phytoplankton cultures, and it's good to see some of my past interns here, Haya Camden, and also light trapping to understand larval-fish recruitment in the Gulf, seasonal variation, population connectivity, what is the population connectivity between here and other areas around the Gulf. With that, thank you very much, ladies and gentlemen, and sorry for talking too much.
Thanks, Willy. Wow, this was a very lively presentation. Thank you so much. All right, keep your question for the end, remember. Now I will welcome Professor Shadi Amin. And this is, I'm very happy to have Shadi here. We've been talking or in contact for some time now, but actually it's the first time we met personally. So very happy that Shadi was able to come here because this research interest is, I think, very unique, and I'm sure that most of you have never thought about what he's studying here in Abu Dhabi. I will let him just go into details, but Shadi has been working for New York University, Abu Dhabi, since 2015. Before that, he was in the US for his PhD and postdoc, studying microbes, bacteria, and their microbial host, microalgae. So microalgae and bacteria are doing some nice relationship, are having some nice relationship, and this is what he's studying since quite some time now. So I'm just gonna leave the floor and welcome Shadi on stage. Thank you.
Thank you. So thank you for the invitation. I'm really excited to be here, and I'll tell you a little bit about what we do in my lab. Let me just try to put the presentation up. All right. So as we've heard Willy talk about, I'm going to kind of move to this side. I think it'll be easier. I work in New York University, Abu Dhabi. I've been there for about 8 years. My lab's name's called the Marine Microbiomics Lab. If you're interested in learning a little bit about what we do, I have a QR code here you can scan. And basically what we study is the role that the microbiome plays in its host's physiology, adaptation, and evolution. And the microbiome is now a really hot topic. And so we study a variety of hosts, phytoplankton, which we'll hear about today. We also study corals and how its microbiome enables it to withstand things like thermal stress and environmental stresses. We also work even outside the marine environment, where we work on the human gut microbiome and its role in metabolism of pharmaceutical drugs, as well as the oral microbiome. So, as we've heard, phytoplankton are extremely important. If you take 2 breaths, one of these breaths have come from the ocean, and the other one have come from all the land plants and forests that exist in the world. So obviously, these organisms are extremely important. They're fascinatingly beautiful, and I'll show you a lot of photos that really inspired me to work on these organisms. But let's first, we're telling you here that half of the oxygen on Earth comes from the ocean, but, don't take our words for it. Let's actually look at data that tells us a little bit about this. So this is a classical paper back from the late 90s that kind of established that fact, that in fact, half the oxygen on Earth and half photosynthesis that occurs in the world occurs in the ocean. So what we're looking here is a world map, and laid upon it is satellite data. So satellites, one thing they're really good at is trying to measure ocean color that reflects off of the surface of Earth. And in this case, right, you can manipulate this measurement from satellites, and you can calculate proxies for things that happen on Earth. For example, chlorophyll. So chlorophyll is the molecule that all photosynthetic organisms have that enables them to do photosynthesis. And using abundance of chlorophyll, you can calculate a parameter that we call net primary productivity. Net primary productivity, just think about it as the excess amount of energy that photosynthetic organisms make that ends up basically fueling all life on our planet, more or less. And so this is basically what we're seeing right here. So the scale kind of describes the more redder the color, the more net primary productivity there is, so the more energy basically available for organisms to be able to thrive and live. And you can see, maybe not surprisingly, that most of that red color lies in the Amazon, right there in South America, in Central Africa, as well as in Southeast Asia. That's not surprising, because this is where most of the forests in the world in the tropics exist. So a lot of photosynthesis, a lot of net primary productivity. And then you see the oceans, when you look at them, they're all in the purple. They're kind of on the other end of that scale. So very low amounts, consistent amounts of net primary productivity throughout the oceans. But of course, the oceans are much, much larger than the continents. And so when you integrate all of this data across the whole globe, what you end up finding is 50 billion tons of carbon dioxide, which is kind of a proxy here for photosynthesis and oxygen production, is removed from the atmosphere every year from the oceans. And about 52 billion is removed by all land plants and forests. So this is where that 50-50 number comes from, basically. And when we look in the oceans and see the breakdown of what kind of organisms that are photosynthetic and how they contribute to this net primary production, we see another classical paper here. So I'm just going to point to this one column, NPP, net primary production. And the total right here is listed at 53. And what you see is most of that is contributed by oceanic and coastal phytoplankton. They literally contribute greater than 90% of all the energy that feeds off basically all life in the ocean. While everything else you see here, coral reef, algae, macroalgae, seagrasses, mangroves, all of these things are so insignificant compared to what phytoplankton can do. And so this is kind of the start of what I kind of want to get into, phytoplankton. So we've heard a little bit, really tell us about phytoplankton. I will get a little bit more details. So there's 2 words from ancient Greek that describes that name, phytoplankton. Phyton, which is plant, and plankton, which is drifting, so drifting or wandering plants. As a biologist, you never say algae or phytoplankton are plants. You get in big trouble. They are not plants by any means. Plants are multicellular. These organisms are single-celled. Also, evolutionarily, they're completely different organisms. They're not related to each other in the slightest. But one thing that's really cool about phytoplankton is they're highly abundant. So every teaspoon of seawater contains, on average, 1,000 phytoplankton cells. So if you take, a empty plastic bottle of Alain and you fill up seawater with it, you have 1 million phytoplankton cells in that bottle. So it's a huge amount of cells. And like plants, as I've said, they have chlorophyll, and so they're able to do photosynthesis, which then converts lights into energy, and in the process removes carbon dioxide from our atmosphere, converting it into organic molecules, and in the process excrete oxygen. And phytoplankton are really fascinating organisms. They're really the inspiration for me to get into this field was really how beautiful they look. And for me, they're kind of at the intersect of beauty, art, and science. They combine all of these three things in really fascinating ways. My lab studies primarily one of these, or two of these groups, but most importantly, this one, which were called diatoms. So diatoms are very unusual organisms. About 20% of all photosynthesis on Earth occurs by these guys. So we said 50%, right, happens in the ocean. 20% of the total on Earth happens because of diatoms. And they have these crazy and beautiful shapes that you can see in a lot of these photos. And in fact, what you're seeing, one of the unique evolutionary mechanisms that diatoms evolved is that they actually built around their cell wall a shell made out of silica. Silica is the same chemical as glass. So literally what we're looking at is a glass house, and inside this brown stuff that you see in the middle is the chlorophyll that does the photosynthesis. And very few organisms on Earth have evolved the ability to mineralize silica in such fashion. And as you can see in some of these micrographs, they're able to manipulate the silica at the nanoscale. So I know of material scientists that devote their whole career on studying diatoms and how they manipulate, silica at this scale, because if you can do this, you would revolutionize the semiconductor industry. You'd go down orders of magnitude lower than what we can do nowadays. And as you can see, some of these cells are crazy shaped. They kind of look like man-made, right? They don't look real, a star, a triangle. And in fact, they inspired people from almost 100 years ago, where we have, during Victorian times, hobbyists that would go in, collect seawater. They would isolate these diatom cells under microscopes, dye them with different dyes, and then organize them in these kind of really cool illustrations. So if you actually go on Google and type Victorian diatom art, you'll find hundreds of these photos all over the internet. And I happen to also be interested in art, so actually I collaborated with people here in the UAE. So the UAE had a pavilion in the Floridi exhibition in the Netherlands in 2022, and they were interested in representing the types of diatoms that exist here in the Arabian Gulf. And so actually I helped them, work out making these plastic molds of diatoms that they molded with glass. And in the end, basically what they've constructed is this really large illustration of diatoms made of glass, because they are, in literal sense, made of glass. Another really interesting group of algae that I want to talk about are known as coccolithophores. Coccolithophores are also another very weird evolutionary event, where these cells evolved something different. They have their cells surrounded by these scales. You can see one of those scales right here, made out of calcium carbonate. So if you're as old as me, calcium carbonate is the same chemical as the chalk that we used to use to write on blackboards. So fascinating. Again, very few organisms can do these type of things, and as you can see, they can still manipulate these things at very fine resolutions, which is really fascinating. And the last lineage of phytoplankton I want to talk about before we move on are called dinoflagellates. So dinoflagellates, maybe they don't look as interesting as diatoms or coccolithophores, but they're also very strange organisms. So the genome of these organisms, of these dinoflagellates, are on average about 15 times bigger than the human genome. So imagine a single-celled organism with a genome 15 times bigger than your own. How is that? Also, they have a very unusual lifestyle, so they tend to do photosynthesis like all algae, but they also tend to eat a lot of bacteria and a lot of other smaller organisms and feed off of that as well. Dinoflagellates are also associated with a lot of harmful algal blooms, and I will talk quite a bit about harmful algal blooms in my talk. They're not the only algae that do this, but they're one of the most important ones that do. And this is one type of those blooms called red tide. You may have heard of that term. Mainly, they grow so much in seawater that they end up changing the water color. And in this case, this species happens to be red, so it gives this red tint to seawater. If you go to Fujairah at night, some times of the year, sometimes even on Saadiyat Beach, you will see also these types of blooms called bioluminescent blooms. These are also dinoflagellate species that when you have waves or you have any kind of movement in the water, maybe a fish, or if you even move your fingers, you'll see light emanating from these cells. So they're really fascinating organisms. So why should we care about phytoplankton besides the fact they make half of the oxygen on Earth? There are three things that I want to kind of point out in this very complex graph, which summarizes a lot of what we know in microbial oceanography. So here is kind of representation of phytoplankton, and there's three processes that are essential for why they matter to life on Earth. One is called grazing. We've heard a little bit about that from Vili. So basically, higher organisms like zooplankton, like krill, copepods, they end up feeding on phytoplankton. That's their natural food. And small fish then feed on these guys, and larger fish feed on the small fish, and so forth until you reach the top predators in the ocean. So basically, the whole marine food web is laid in the foundation of phytoplankton. So without phytoplankton, there's no fish in MENA market. Another thing that's very important about phytoplankton is when the cells die, like everything else on planet Earth, they basically get sticky, they aggregate together, and they form larger particles that then settle down the water column fast. And when they do, remember that they do photosynthesis, so they capture CO2 from the atmosphere and they convert it into organic molecules. All these organic molecules with these dead cells come in and they settle all the way down at the bottom of the ocean to the seafloor, where they're buried forever, supposedly. And that's a much more efficient mechanism of removing CO2 from our atmosphere compared to a tree, right? A tree, you can plant as many trees as you want. They take this, again, CO2, and they deposit it into wood in their trunk. But a tree is eventually going to die, or it's going to get cut to be used for some production of some kind, or it's going to get burnt. And all of this CO2 goes right back into the atmosphere. So it's not a really efficient mechanism of removing CO2 from the atmosphere compared to what phytoplankton can do. And the last thing that they do that's very important, and this is a lot of the stuff that we do in my lab, is this third one, which is the production of organic molecules. So when they do photosynthesis, they take this carbon dioxide, they make organic molecules, and a lot of these organic molecules are secreted outside of the cell. They leach it out. And for a long time, oceanographers thought, that's kind of weird. Why would they invest energy into making all of these molecules and then just spit them out? And back then, they thought, maybe these cells are leaky, they're getting exposed to too much light, and they don't know what to do with all of these molecules, so they're just getting rid of them. But we now know that this secretion is very intentional, and the reason for that is these phytoplankton cannot survive on their own in the oceans. The ocean is a very turbulent environment. It has all kinds of nutrients that go up and down, and vitamins and metals that are all required for the growth of phytoplankton, and they cannot just get everything on their own by themselves. And so what they do, like a lot of higher organisms, they associate with a bunch of bacteria. And bacteria are very good at getting and providing a lot of different things, not just for phytoplankton, for us, for corals, for plants, for pretty much every higher organism that live on Earth. And so in this case, we know from a lot of work that we've pioneered in my lab that these algae, or phytoplankton, they produce a lot of molecules to attract bacteria. I'm representing this here by these blue arrows. So these are things like sugars and amino acids that let bacteria come close to them. And once they do, they then secrete a bunch of signaling molecules. This is these red arrows right here. And these signaling molecules have a very important role in selecting for the right type of bacteria. So if you're under some environment where you need a specific vitamin, typically vitamins come from bacteria. So what you're going to do is you're going to secrete signaling molecules that's going to attract bacteria that make that vitamin for you. And once you do that, these bacteria come in and colonize this microenvironment around the cell, provide it with the vitamin, and the bacteria feed off of these organic molecules that the phytoplankton make. And at the same time, the signaling molecules fend off bad bacteria, parasitic bacteria that otherwise would come in and kill these phytoplankton cells. And we know that when phytoplankton cells become, they age, they're close to dying, they're unable to do these mechanisms of secreting molecules and signaling molecules to achieve this very beneficial microbiome. And so what they end up having is you'd have more of these parasitic bacteria that eventually take over, and then they kill the cell. And these tiny interactions, these interactions are really at the micro scale between a single phytoplankton cell and a few bacterial cells. We now know that they're extremely important on the ecosystem level. So these are some of the very important processes that these types of interactions catalyze. And so, for example, first one right here, ecosystem productivity. So whether or not you're going to have very healthy ecosystem with a lot of fish, believe it or not, depends on whether a phytoplankton cell has the right type of bacteria that's giving it the right type of chemical that it needs. If you have these types of bacteria, fantastic, phytoplankton grew well, zooplankton eat them, other fish eat them, you got a very healthy ecosystem. If you don't have these types of bacteria, the whole thing breaks down, you don't have a very healthy ecosystem. Same thing happens when you have something like harmful algal blooms. So with harmful algal blooms, it's typically associated with toxins. So these algae produce toxins, and those toxins harm mammals and humans. And we know, for reasons that we still don't understand, that these bacteria control or regulate the production of these toxins. We don't know how, but we know that they do. When I told you about aggregation and whether carbon gets driven down to the bottom of the ocean or it gets recycled back into the atmosphere, this carbon cycling is highly controlled by these bacteria because they're able to enhance the aggregation of phytoplankton cells or inhibit it. So again, how does it go in terms of carbon going up to the atmosphere or down? Has to do with tiny bacteria that live around these phytoplankton cells. And even when we talk about clouds, so cloud seeding is a big thing here, right? Because we need more rain. So it turns out that these interactions produce copious amounts of a gas known as dimethyl sulfide. And dimethyl sulfide, or DMS, goes up in the atmosphere and acts as a cloud nucleating agent. And so you can think of it as kind of a natural form of cloud seeding that happens, again, because of very, very tiny interactions that happen between these different organisms. So, of course, this is all great, but of course, as human populations increase, as we have a lot of food that we have to produce, a lot of agriculture and fertilizers, that leads to runoffs from coastal areas into seawater. You got sewage and wastewater that sometimes is dumped into coastal areas. A lot of these things introduce a lot of nutrients into seawater. And those nutrients, along with that microbiome, help these phytoplankton to divide and grow very fast. And so that's when we end up having these harmful algal blooms. Some of them are green, like these. Often, these are cyanobacteria, so actually types of phytoplankton that are actually bacteria. You got ones that are red, like the ones we've seen before with dinoflagellates. And if you go to places like Florida, where these blooms happen all the time, every single year, you will have a bloom. Can last a couple of months, can last a whole year. And we've been studying this for quite some time in the US and the Gulf of Mexico. And it's very often to see these types of signs where, these types of things can cause eye or skin irritation because of the toxins that the algae produce. You can even have symptoms similar to asthma if you inhale aerosols that have these toxins. And of course, it's extremely bad to be fishing or eating any kind of fish or shellfish or so forth from there, because these organisms are accumulating more and more of these phytoplankton cells along with the toxin. So if you eat that, you're getting a super high dose of the toxin, much higher than let's say, if you're swimming and you just swallow a little bit of water. So obviously, they have major impacts on marine ecosystems. You got sometimes a lot of fish kills that happen because of these blooms. And you may think that they're really localized in different small beaches here and there, but they're actually quite large. So these are satellite images. This is not a calculated image. This is a real photo from a satellite. And you can see how large that bloom is. This is from the Norwegian coast. You can see it in the South Atlantic, Black Sea. You can keep going on, right? The Irish coast. And if you Google satellite harmful algal blooms, you'll find thousands of these blooms that occur worldwide. And it's believed that because of climate change, that there is an increasing incidence of these blooms worldwide, which obviously has really bad implications on fisheries and local economies. And the Arabian Gulf is really not any different from the rest of the world in that sense. So we get a lot of blooms in here. And in fact, the Gulf is very interesting and odd in some ways. So it's the hottest body of water on Earth during the summer. And when my lab a few years ago, maybe a little bit before COVID, started going into the Arabian Gulf and sampling water and kind of sequencing DNA from there to try to understand what kind of microbes exist in there, we were really surprised to find out that dinoflagellates, those algae that cause all of these blooms, are actually the most abundant group of phytoplankton that we have. And so this is just some of the data that we have. This kind of red pinkish color represents dinoflagellates, along with a lot of other eukaryotic type cells. So these are phytoplankton as well as non-phytoplankton cells. But by far, dinoflagellates are the biggest phytoplankton group, and they're averaging about 50 to 60% of the phytoplankton population, which is quite significant. And as an oceanographer, it's surprising because we typically imagine places like the Arabian Gulf, kind of an estuary, kind of shallow, to be dominated by diatoms. To see a place like that dominated by dinoflagellates is quite odd. And we still don't understand why, but we're doing quite a bit of work to try to understand this. Of course, that's problematic because if you have a lot of dinoflagellates, by default, you're going to end up having these HABs that have toxins. And I want to just give you one example. This happened quite a bit back in 2008 and 2009. This was the biggest harmful algal bloom that happened in the Arabian Gulf since records have been started. And so this is started right here in the Gulf of Oman and then entered the Gulf, and basically within 3 months, right, from October to January, it had spread throughout the whole Gulf. And it lasted for about 9 months, which is a pretty long time. It was instigated by this one species of dinoflagellates right here. And it caused a lot of havoc because, of course, fishery has to stop, right? It wasn't very safe to swim in the water. Water desalination plant had to shut down because they cannot operate when you have these blooms. And in fact, in the UAE alone, there was a greater than $100,000 loss per day during those 9 months because of the inability to produce fresh water. So, big economic problem. Now, naturally, what we do to deal with this is you try to do two things. You try to monitor, so you try to kind of go out, collect water sample on a regular basis to look for these blooms and try to see if they do happen. Then you can close beaches, you can regulate what you're going to do with desalination plants and so forth. But the other thing you can do is to try to predict. So you can use, for example, satellite data, like I've shown you with chlorophyll, and try to see if there's a bloom that's going to happen and then take action before it spreads out. However, in the Gulf, this is quite problematic because I'm showing you right here a map of the bathymetry of the Gulf, and the Gulf is extremely shallow, unusually shallow. In fact, here's the coast of the UAE, and basically from Abu Dhabi, where we are right here, all the way down to almost half the Gulf, the depth is about 10 to 15 meters, which is extremely shallow. I mean, estuaries are typically even deeper than that. And it doesn't really get very deep until you get to the Iranian coast, and even then it's about 100 to 130 meters. So still, relatively speaking, compared to the oceans where it's 10,000 meters, 130 meters is nothing. And that creates a problem because what happens when you have a shallow body of water is that the light that's reflected off the surface of the water that satellites use to measure chlorophyll, for example, gets mugged up by reflection from the bottom sediments. So now your satellite approximation of chlorophyll is no more accurate. And you can even see this here in this example where we know that this bloom spread out throughout the whole Gulf. But as you can see, satellite data is telling us that the bloom is right here, which is the deepest part of the Gulf. There's nothing on the UAE side, there's nothing up here, and those are very shallow areas, so satellite data is just not reliable. So that becomes a problem, and we know that these blooms happen all the time. These are just some examples from, if you read the National, for example, where they sometimes report these types of blooms. So I'll tell you very briefly about one case study that we've been doing right here in the neighborhood in Yas Bay. So this is a map of Abu Dhabi, and right here, this is Yas Island, and in the south of Yas Island, we have Yas Bay, which is this part right here. This is the commercial part of Yas Bay. I presume most of you have probably been there. And down here, you have a whole bunch of residential areas. This is Al Zaina, Al Munira. Anyone here lives in any of these places? Al Bandar, Al Raha, okay, some people. Okay, so you probably know what I'm going to be talking about now, right? So we were alerted about a year ago, I believe around April of last year, by a colleague who works at NYU with me, that, hey, I live in Alzena and the water smells so bad, it's so muggy. You're an oceanographer, can you just tell us what's happening? So, okay, scoop some bottle of water, let's see what's in there. And this is kind of the stuff that residents in that area are experiencing on semi-daily basis. It's obviously not very nice if you have a beach that you want to swim in. We looked at the first sample we took at, and we found basically a massive diatom bloom. So all of these things are diatoms, as you can see right here. And that was kind of interesting. But one of the things that threw us off or kind of scared us is all of these cells that look like needles. You see a whole bunch of these needle-like cells. These are this particular group of diatom species known as Pseudonychia. And Pseudonychia species, there's multiple species that exist within these groups, but a few of them actually produce a neurotoxin known as domoic acid. And that's a toxin that basically targets your neurons. And so if you ingest, for example, fish from a place like that, you get enough domoic acid, it could lead to a lot of bad things, including death. And so what we've done is then, since we do a lot of chemistry in my lab as well as biology, we have an instrument called a mass spectrometer that you can then take an environmental sample and look for molecules in there. And lo and behold, we were able to find domoic acid. And so at that time, we've worked with the environmental agency Abu Dhabi. At the time, they actually ended up shutting down the beach for quite some time. And we kept monitoring this until eventually we were able to see that domoic acid disappeared, which was good. And so in kind of our work on Yas Bay, we've been actually collecting samples. As you see here, all these little yellow things are kind of stations that we target every single week. And the person who really is doing most of this work is Julia Bibre in my lab, who's sitting right here. So she goes out every week, collects water samples from each one of those stations, brings all of those samples back to the lab where we do a lot of analysis on them to see what's happening with this bloom. Do we see toxins? What's happening with the nutrients in there? What might be giving rise to this crazy bloom? Note, this is from April 2023. We're in March now, 2024, and there is still a bloom. It's a very long-term bloom. And in fact, I've already mentioned to you, so the Environmental Agency Abu Dhabi has actually been quite a good partner in a lot of the stuff that we're doing there. And I'm going to show you just a little bit of data that we've been gathering across this whole year from there. And so this is from one of the stations that we collect. Here we're looking at nutrients that typically are associated with the occurrence of these types of blooms. And what you typically see is there's not really much happening with most of these nutrients except with the black one right here, which is nitrate. And you could see with nitrate, right, it was very high in April when we first detected this bloom. It kind of crashed, then it had a big spike, crashed, another small spike, and you get all of these little spikes, and now it's shooting quite high again. So this is indicative of some kind of point source infusion of nitrate that's coming from somewhere. Don't really know where, but this keeps happening over and over and over throughout the year. We can look at the phytoplankton populations that exist in these blooms, and this is again from a station that we collect from. And what you typically see, here's the major groups of phytoplankton that we look at. Diatoms in blue are by far the dominant thing, which makes sense because this is what we tend to see under the microscope. And so what we can do is then delve further into diatom population and look what species exist in there. And you see two colors, blue and purple. So the blue is typically this one group of species called Skeletonema. They tend to not be toxic, so it's not really something that would be too worrying. But the other problem is these purple guys, because those guys are very consistently happening throughout the whole year, these Pseudonychia species. Now, not all of them are toxigenic. Some are not, which kind of complicates things because that means you have to always be looking for the toxin to see if it's there or it's not there. And that's basically what we've been doing. So as I've shown you before, this is one of the samples where we detected domoic acid. And then as we went down into September and beyond, we haven't been able to detect it, which is great. So that makes things good. And one article that I actually found while I was making this presentation, this is from 2017, an article in The National, where actually residents in the area were complaining about sewage leaking into the water. And in fact, during our collections, we sometimes notice pipes that are pushing a lot of water. So it's something that's worth investigating and seeing if this is indeed the source of nitrate or not. And maybe the last thing I want to show you, so obviously our ultimate goal is to be able to look at all of these blooms and monitor them to try to eventually predict them so we don't have to actually go and measure them all the time, because that's obviously very costly and time consuming. But in order to do any kind of prediction, you need a lot of data. And that's a big problem in the region right here. Here's the Arabian Gulf. I always say this when I talk about the Arabian Gulf. We know more about the Southern Ocean around Antarctica than what we know about the Arabian Gulf. And that's shocking because Antarctica doesn't have any permanent human populations around it. Yet there's a lot of research that's interested in the Southern Ocean. There's a lot of people that live around the Arabian Gulf, yet we don't know much about it. In fact, throughout its history, there's only been 6 research oceanographic cruises in the Gulf. They're all mapped out right here in this graph. That's almost nothing. And in fact, you could see at the dates, 1965, the latest one was in 2000, 24 years ago, a long time ago. And so my lab is part of a research center in NYU Abu Dhabi called Mubadala Axis. And so our goal is to kind of gather as much data as possible of the Arabian Gulf in order to be able to predict future changes. And so in the past 9 months alone, we were able to conduct 4 large research cruises. So, we're that close to actually doubling what's already been done over the course of 80 years or so in the Arabian Gulf. And so this is, for example, one of our ship tracks where we go out. If you notice right here, one of these little speck islands is Abu Dhabi, and so we're going pretty much almost along the whole UAE coast, collecting water from all of these places. We also do a lot of small day-long sampling trips that we also use to build a transect. And we also deploy a bunch of networks of buoys and other remote instruments that basically gather physical and chemical and biological data and relay that information to us live. And so what we've done so far is we're already starting to find some really cool, interesting findings just simply because there hasn't been much known about the Arabian Gulf. And so from some of our research cruises, we already know that the Gulf is extremely oligotrophic. Oligotrophic means that it doesn't have much nutrients. And in fact, that's what these depth profiles, so here each one of those is depth versus the concentration of different nutrients, and all of them are extremely low. These are not the type of nutrient levels that you would expect a bloom to happen in. So like Yes Bay, my hypothesis is if you have a bloom happening in the Gulf, it's probably happening because of a sudden input of nutrients from somewhere. It could be either coming from the outside, from the Gulf of Oman, or it's coming from the coast somewhere. And another thing that we're trying to do is use NASA's new satellite. So last month, NASA released a new satellite called PACE, stands for Plankton, Aerosol, Cloud, Ocean Ecosystem. And PACE is fantastic because they have very broad bandwidth that enables you to measure a lot more color than any satellite that existed so far around the planet. And so with this type of satellite data, you can actually look at ocean color and you can tell which type of groups of phytoplankton actually exist rather than just measuring chlorophyll in general. But we're also hoping to be able to use that to get over the problem of the shallowness of the Gulf. So around the coastal area of the UAE, can we actually use this very broad bandwidth of color to subtract or kind of at least remove the problems that are coming from the interference from the sediment? And finally, our ultimate goal from all of this data that we're collecting, from ships, from little boats, from satellites, from buoys, and so forth, is to develop biogeochemical models that basically tell us, as global warming happens, and as we basically have a more dire consequences of climate change, what is going to happen to the Gulf? What's going to happen to the fisheries? What can happen to the harmful algal blooms? And all of these very important parts of the ecosystem that obviously are very important to the UAE as well as other nations in the Gulf. And with that, I would like to acknowledge the people. I'm standing up here talking, but really everything that I've shown you right here was done by other people, very talented and amazing people in my lab who've really done a lot of amazing work. I'd like mainly to acknowledge Julia, who's really done a lot of the work on Yes Bay as well as the cruises, as well as Srijit, Salah, and Carly, who's also been instrumental in a lot of this work. Also, my collaborators at NYU-ED as part of the Mubadala Access Center, Francesco, who's also here, as well as other people at NYU, the Environmental Agency Abu Dhabi, and particularly Sarah, Lamia, and Sultan, And finally, the Jae-woon and Ocean X captain and crew who helped us with a lot of the research cruises that we've conducted, and they're amazing ships. And also thanking our funding agencies for support of this science. And thank you all for your attention.
Thank you so much. Wow, this is, yes, a lot of, I'm excited about that. A lot of new data to analyze. Time for question. So if we, yeah, Vili, come over. And you can start raising your hands. We'll come over with a microphone. Great. All right, first question. Oh, I'm in the middle. Ooh. All right, Leila, over there.
Hi, thank you also, both of you, so much for your speeches. I wanted to know if, because of pollution and climate change, adaptation of photoplanktons would change throughout time and how they would change.
Do you want to start? Shadi, go. It's working.
Louder. Can you all hear me? Okay. Oh, that's working. All right. So obviously adaptation is very important, and especially for microbes. Like when we talk about corals, adaptation is a problem because when we're talking about global warming, the temperature is rising too fast. Corals don't divide very fast compared to things like microbes. But nonetheless, we're talking about a lot of changes. So with the ocean, there's the increase in temperature. That's one big problem. But the other one is ocean acidification, which a lot of people may not have heard of. But as we increase CO2 in the atmosphere, a lot of this CO2 is actually dissolving in water and has an equilibrium between air and water. And when it dissolves in water, it becomes an acid. It becomes carbonic acid. And so what happens is the pH of the water is actually reducing and it's becoming more acidic. And that's causing a lot of problems for a lot of microbes. And for example, when we talk, I was showing you coccolithophores. Coccolithophore has a calcium carbonate shell. If you expose them to a lower pH, it dissolves. And so they're no more able to live their normal lifestyle. And so we still don't know a lot about how these microbes are going to adapt. If I have to make a kind of an educated guess, is that we're going to have a lot of big changes in populations in some places, especially here in the Arabian Gulf, where it's very, very hot. But I don't think that these organisms are going to go away. I'm pretty sure there's going to be quite a bit that are going to be able to survive and adapt. But I think there's going to be a shift in populations. And that shift perhaps is a little bit worrying because it could have an impact on fisheries, because the grazers that are feeding on phytoplankton are not just feeding equally on everyone. They tend to prefer certain types of species and groups of algae. And so if you have a shift in the populations, you may affect how the grazers are feeding upon them, which means you're going to affect the whole marine food web.
Yeah, just do you want to add to that? Because this is one of the things that you're looking at when you have small fish.
In terms of the Gulf, I have not as much experience here, but in terms of coral reefs in the Pacific, I've worked in projects that have involved management of the cascade effect, and in terms of holistic approaches, they call it from ridge to reef, where unsustainable farming practices are carried out, and that leads to the cascading effects that are felt in the coastline areas. This leads to overnutrification, it leads to microalgal and all the disruption in the food web. And as has been alluded to, this leads to disappearance of key players in the food chain, leading to corals being overtaken by microalgae, and then you have a gap in the food chain and then collapse in the fisheries, which is what we're looking at. And most of the struggle in many other places with large riverine systems is having the approach of managing from the ridge all the way to the reef, as they're usually having different judiciary bodies that are involved and having them get in line This is usually the challenge, but in the Gulf, I hope to work a lot more within UAD, and hopefully we can share experiences.
And just to add to that, I guess it's really the shift in population. It might be at the end of the day maybe the same amount of photosynthesis or the same amount of, but if it's not produced by the same cells or organisms, then it causes a problem exactly that. It's just like when I take the example of the ciliates that are just a component maybe above the phytoplankton, then they will not grow the same, and then there will be an impact on the small larvae of anchovies or the small larvae of sardines and so on and so forth. So it's, yes, just having an impact on the phytoplankton is a massive impact for everything else in the ecosystem. All right, another question. Camden?
Hello, yes. This is for Dr. Villi. You mentioned how phytoplankton decrease in depth or increased depth during the day or during lunar cycles where it's a full moon or a bright moon. What mechanism or organelle enables them to do that?
Okay. Wow. Well, in terms of my experience, fish larvae that are not really phytoplankton, but more zooplankton. The reason why they avoid more full moon nights is predator avoidance. This has been what many papers have done, and what we found in our study is that fish larvae prefer to remain on the bottom, further away from settlement sites or where they will end up. And then on new moon nights, they will then transition in. There's also secondary areas, nursery habitats, where they would go onto like mangroves or seagrass beds, hang out there for a little bit more until they feel that it's a little bit more, they're a little bit bigger, and they can swim to evade predators before they go on to their final habitat. So it's usually around predator avoidance. This is the usual, what we've seen in many papers as well, has been why they choose to avoid full moon nights. And it makes sense also. Apart from that, other adaptations are that they are transparent with silver abdomens, so that they're not easily seen through, in contrast if they are above the water and the predator is below. These are some adaptations that the larvae undergo to avoid basically getting eaten. I think I should answer that one.
And just to come back to your first point, I guess the vertical migration, the dual vertical migration is mostly zooplankton. The phytoplankton will stay where the light is, right?
Yeah, so phytoplankton cannot swim against the current. They're too small.
That was what I was confused on, is that since they're so small and unable to move against or swim on their own, that how are they moving in depth? Yeah, since it's zooplankton, not phytoplankton, then that makes sense.
So the belief is that some phytoplankton can actually play with their buoyancy, and that enables them to kind of move on them. They're not really moving, but they just, with manipulating buoyancy, they can just go up and down. Some of them are motile. So like dinoflagellates, for example, they actually tend to swim quite fast. But in general, very small particles like this in seawater are unable, they're basically moving with a parcel of water. They're unable to jump out of it, if you will.
Thank you.
Another question? Yeah.
Yes, hello, and I think it's very related to the previous question. Billy, you mentioned that the fish larvae, especially, they drift in the open ocean and somehow they come back to the reef. Mm-hmm. Do we have an understanding what is the mechanism? How, I mean, at the end they need to have a memory. So, do we know how this works?
It was covered a lot by one of our first talks and science talks. So there are many cues. Some sound, reef sounds, chemical. chemical cues. And they've discovered, for example, in our study, high volcanic island that was more rare had river systems. And some of the cues were from the freshwater outflow. This could have explained why we got a more diverse catch in the high volcanic mountain compared to the atolls. So there are multiple, one of the more famous is out of the University of Bristol on soundscapes. I believe there's a documentary out there on that reef fish notice some sounds, the clicking of crabs on reef heads. And this could act as a cue, attract them for some fish that are only associated with a certain type of coral, species of coral. Why is this fish only associated with Pocilopora or Acropora? It's up to this scale of attraction to a certain species. So there's still a lot of question marks on this in this part of work in terms of cues and settlement of fish in the transition from juvenile to adult substrate or benthic associated life stage. But yeah, it's a pretty interesting field.
Thanks. Question here.
Do you want to address that? Fish.
Whale shark. I don't know. I don't have any. I wouldn't speak on anything that I don't have any knowledge on whale sharks and tied into.
Yeah, I mean, I wouldn't know how sharks are figuring out, which plankton they prefer and how they get that's out of my comfort zone.
No, but I think it needs. I think maybe you know which plankton attracts them here in the area, or which plankton is most frequent at the times that we find whale sharks. I thought there's somehow an interaction that you may know.
Well, here's a question. Maybe you guys know. What does whale sharks eat? Do they eat
krill,
phytoplankton?
Not phytoplankton. Yeah, but mixed. It's a opportunistic sort of a feeding, ram feeding. To tie to a certain species and make it like an indicator as for migratory species is a little bit. I'm not out of my field also. Sorry.
I think just to answer your question, we need more data. And this is one of the things that Shadi is doing.
No,
it's true. It's true.
We always need more data.
Because we need to have the data of what is in the water when the whale sharks are here in the water, and then you will see if there is any relationship between the location of the whale shark at the time and the quality of the water or whatever is in the water.
And actually, speaking to that, we still don't really know the plankton diversity in the summer here, because no one has managed to do anything in the summer because it's just too hot. So everybody's doing stuff in winters usually. And so you have these big gaps in time where we really don't know.
But the whale sharks are coming also more in the winter season, not in the summer season as far as I know. So it's not the summer. I thought it's known. So maybe next time when the whale sharks are here, you need to go out and measure.
We'll sample. Yeah,
we need to develop
I guess there's room for baseline surveys to be done on that.
Thanks, Claudia. Another question over there. Yeah.
For the presentation earlier. It's really interesting, some really good stuff. I had a question on the carbon sinking, because there's a huge amount of research and money goes into planting trees and protecting rainforests, and I was wondering if there was any projects in phase for seeding the phytoplankton that's most effective, or trying to ensure the conditions are protected for those phytoplankton to do their job effectively.
Yeah, that's a very good point. Actually, I didn't have time to show this, but one of the findings that was very fascinating back in the 80s was when oceanographers found out that there's these large areas of the ocean, huge areas of the ocean, where you have very little phytoplankton, yet you have all of these nutrients that typically phytoplankton need to grow. And they call these areas high nutrient, low chlorophyll regions, HNLC. It's very common in oceanography to hear about that. And it took years to find out that what was limiting all these phytoplankton from growing was iron, that there's not enough iron, and they need iron. Everything on Earth needs iron, and for them, photosynthesis relies on iron. And so one of the really cool experiments they've done was to take big barrels of iron, dump it down the back of a ship, and just cruise the ship as you dump the iron. And then you use satellites to look at what happens to chlorophyll. And as you can see, a ship is moving, and lo and behold, a week later, there's a big trail of chlorophyll that's following that ship and lasts for maybe a month or so, and then it disappears. So that established that large areas of the oceans are iron limited, and they're limiting mainly phytoplankton. And so nowadays, as of at least before COVID, there were commercial companies in Canada, for example, that do this thing called ocean seeding, which is you go out to international waters on big ships, you have tons of iron, literally, and you're dumping that iron down, inducing these artificial big-scale algal blooms. And the idea is, if you do that, you would capture a lot of CO2, eventually the bloom dies, sinks to the bottom, you've removed a whole ton of CO2. Right now, it's still quite controversial because some of the research that was done on it established that, well, sure, you do fix a lot more or remove a lot of CO2 from the atmosphere, but what else do? You increase the activity of that microbiome that exists with these algae. So what does that mean is as the algae are taking up carbon and secreting it to the bacteria are taking that carbon and respiring back into CO2. And so you get a feedback cycle that just increases in magnitude. So you take more CO2, you release more CO2. And we still haven't figured out how do you reduce that release of CO2 and instead increase the aggregation. But that's a very good question. But a lot of people are interested in doing that because it is much more promising than just planting trees.
Thank you.
Thanks. We have a question here.
Yes, so my question is about what's the expected rates for dangerous algae blooms in the future? And is there any preventative measures for them?
Question for you.
Wow, very good question. I do not know the answer to your first question. It's a very good question. It's believed that these blooms are increasing in frequency. We don't know the reason, but obviously the fact that we have increasing human populations is probably one of the main reasons why this is happening. In terms of prevention, unfortunately, there's been a lot of technologies that have been tried to kind of tackle that. And so, for example, there have been patents where people put zeolites, some kind of benign particles in seawater that would basically come in and settle. And as they settle, they just drag a lot of these algae down to the bottom. But this doesn't seem to work very well. Other people have looked at manipulating circulation to deal with these algae. Also only works in very specific areas, so a place like Yas Bay maybe, but in an open kind of ocean or Gulf, environment that wouldn't be very easily. So unfortunately, we actually don't have a solution for these blooms, and that's why throughout the world, all governments, what they do, you monitor and you predict. And when a bloom is happening, you shut down the beach, prevent people from swimming, you shut down fisheries, and that's about it.
What about any way to displace the species of plankton?
So that is dangerous because if you think about an ecosystem like that with thousands of species and maybe, one thing is blooming, if you come in and you say, I'm going to target this one species, I'm going to wipe it out, first of all, you'd have to have a very directional and specific method to do that. And unfortunately, most of the things that we have, let's say adding a chemical or adding a biological agent or whatever it is, it's going to affect a lot of other species. And what that means is you're going to disrupt the whole ecosystem without really knowing how it's going to get disrupted. And that becomes a very gray area that no one wants to touch. You don't want to mess with an ecosystem and hope that it's going to go well.
Okay, thank you.
Thank you. Another question over there? Adam?
How do planktons know where to go and what to do if they don't have a brain?
Whoa, this is quite a question. Thank you.
Yeah.
Really?
Yeah, that's a very good question. It's a similar answer to the one that I had given earlier. So, plankton, as I mentioned, have 6 to 8 weeks out in the open ocean. They, depending on the species, they come back still clear, transparent. Some are translucent. They fully develop the notochord flexes, the spinal cord, and all this happens while they're out in the open ocean just before they cross. So there are currently many researchers who are trying to answer that question. How do they know when to come back, where to go? So there are multiple cues or attractants, you can say, that attract them towards a specific coral reef or a specific type of coral. So these can be in the form of chemicals in the water or sound, as I mentioned. They can also be visual. As they come over the reef, their eyes have developed to be able to recognize also the type of corals that they would like to be, that their parents or where they spawned, where they would ideally like to be transitioning into a juvenile and also later into an adult. So scientists are saying that it could be singular cue or it could be a combination of sight, sound, smell that could be driving this fish to their final grow-out phase in terms of a reef or a species of coral.
Nice. And then going back to phytoplankton, how do they know which molecule to produce to attract which bacteria?
Yeah, so it all relies on chemical signaling. So I teach a course at NYU called Communication from Bacteria to Humans, and we talk about how do all forms of life communicate. And surprisingly, even bacteria, they talk to each other. They have a language that's made up of small molecules, and that's how they can talk to each other and tell each other messages, basically. And that's what happens between these phytoplankton and their bacteria. They talk to each other via small chemicals that tell each other to behave in a specific way. For example, in my lab, we found that when these phytoplankton want to attract a bacterium that benefits them, they secrete a very specific molecule that tells the bacteria you need to attach. And if it's a bacterium they don't want, they secrete a different molecule that tells this bacterium, don't attach, we don't want you here. So a lot of this stuff happens via these very small chemicals.
Amazing. It's just, I can see Adam is just like still thinking about it. It doesn't make sense, but yes.
Yeah, we also conduct some sort of choice chambers where you have different light colors. You put a fish, and then we see which is attracted to red or is attracted to blue. And we found that some species, Chrysiptera, that we worked on, actively chose a singular color. So it could also be very strongly visual as well for most of these larvae.
All right. We'll take one more question and then...
Hi, thank you so much for the feedback from Yas Bay. I am actually based in Yas Bay. I normally realize that in some, within the year, the water changes color. And also we have seaweed blooming depending on the time of the year. As you have said that you are still testing the waters, we normally have water sports taking place, and us as divers, also we have dive missions underwater. How can you, explain if it's dangerous to go underwater, or is it okay at the moment? Thank you.
Yeah, so that's a good question. It's generally safe to go to the water. So the main problem is generally the toxin. If there's a toxin, that creates a problem. There's multiple ways it can create a problem. First of all, fishing is definitely bad if there's a toxin. Definitely do not fish. And in fact, I would just say, just don't fish from Yas Bay. I know some people do. Probably not a good idea when there's a bloom, whether there's a toxin or not. The second thing is if the toxin gets in aerosols and then you breathe it. That's probably not a big problem in Yas Bay because there's not a lot of waves and wind to drive that. So I think the main thing is really eating seafood from that area. If you dive, the chances that you're going to ingest so much water with so much algae that make the toxin is not that high. So I wouldn't be worried too much about that. I would just be worried about fishing.
Okay, thank you so much. Because at the moment, the marina doesn't allow fishing, so that's out of
good.
the question.
I've seen people who do fish on the side, probably just on their own, but... Yeah.
Also on El Raha, there are signs that say don't fish here. And those signs are up for a reason because I guess the government also put them up for a reason so that people don't get the poisoning. So please don't fish.
Thank you.
All right. We're going to close. It's late. It was so interesting. Thank you, Billy. Thank you, Shadi. I hope you enjoy the night, and I hope I will be able to schedule more talks on plankton, if you allow me. Next talk after Ramadan, after Eid, it will be with a colleague of yours, Dr. John Burt, and we will be talking about the new book that is out, and I'm guessing you part of the authors, about the natural history of the Emirates. So come over, and I think it's 25th of April, but we'll just keep on looking the website and you will see the date. Thank you everyone for coming tonight.